Hello and welcome to this short video on how to answer a long answer question on genetic engineering. The question that we're going to focus on is, in some humans, they suffer from diabetes. This is caused by an inability to make the hormone insulin. Describe how human insulin can be transferred into a bacterium so that it can make insulin. As this is a long answer question, it will usually be out of 6 marks, so you must remember to make at least 6 points. You must remember to write in full sentences and pay careful attention to using keywords and grammar correctly. The first point in any question along these lines that needs to be made is that you need to extract the gene out of the cell. So before doing that, the cell that you need to extract it out of needs to be identified. In most cases, if we're speaking about insulin, it will be a beta cell from the pancreas. However, if the question on genetic engineering is talking about a different situation, all you need to do is state the particular area or organism that the cell is coming out of. You might also want to mention what the function of the cell is at this point. The second stage is to talk about extracting the chromosome or DNA from the nucleus of that cell. You may wish to mention that the nucleus or the nuclear membrane is broken up using a salt solution and being warmed up to make extracting the DNA easier. Once you've extracted the DNA, the specific gene on that string of DNA needs to be identified and cut out using restriction enzymes. You may wish to state what the gene will do again, and for some exam boards you may need to make reference to the enzyme which is cutting the DNA, leaving sticky ends which allow the DNA piece that is cut out to be stuck into another piece of DNA more easily. The next stage is to talk about a plasmid being extracted from a bacteria. A plasmid is a circular piece of DNA that the bacteria uses to transfer genetic information. In this specific question, we're talking about genetically modifying bacteria, but in another different situation, you may want to talk about taking the DNA out of that specific organism. Once the plasmid has been extracted, it is cut open using restriction enzymes. The restriction enzymes will cut the plasmid in the same position as it cuts out the gene. Again, you may want to make reference to it leaving sticky ends to allow the gene to be stuck into it more easily. The gene for insulin is then stuck into the plasmid using a different enzyme. Again, make reference to the fact that the sticky ends are connecting to each other. The plasmid is then reinserted back into the bacteria, so now the bacteria has the gene for producing human insulin. To allow the plasmid to enter the bacterium more easily, we warm them up to make the membrane more permeable. The bacteria is then grown in a fermenter at pH 7 at 35 degrees with the correct level of nutrients and oxygen. These conditions are to allow optimum bacterial respiration, which increases bacterial growth. As the bacteria grows, it produces insulin, which can then be extracted and purified. If the question is talking about some other type of organism, then you need to make reference to it at the end. Below there is a link where you can download a copy of this 6-mark question so you can now have a go at it. Feel free to rewind the video and pause it at any point to help you answer the question. Good luck with your exams.